Good evening. Yes, good evening, everyone. Now, as promised, today we'll be going for the embryology. So, few emails I received. One was about the exam. Yes, tomorrow there will be a test day, right? And I think 40 questions should be enough, right? 40 questions. Okay. So we'll be having about 40 questions, and I'll be giving, say, 30 seconds. I think 30 seconds should be more than enough, right? Because say if we spend more time, so then there will be less time available for the discussion. So let's keep it, say, 45 seconds, right? So I'll keep exactly 45 seconds to answer every question, and once they are done, we'll be going for the discussion on it. In that poll, I was very happy when many of you have answered like that. Even if we have not read, yes, we'll be happily attending the test, and definitely it's my promise that you'll be enjoying it so much, right? Okay, 40 is good. Great. Timing of the test, well, timing of the test is same time, 7:30, right? It would be 7:30 p.m. IST, right? Same time. In fact, every day, right, from Monday to Saturday, we'll be meeting at this time only. So there is no change. And as soon as we'll complete the test, right, tomorrow I'll be generating all the links for the next week, right? Okay. So good evening, and let's start with our today's session, right? That's the brief introduction. Okay, and let's start. Okay. Now this entire process, say deliberately, I have selected this embryology. Because whenever we study, say, brain, whenever we study abdomen, every time it will definitely come that, okay, it developed from this mesoderm and then it developed from this endoderm, ectoderm, and this is how at times, say, this entire process, it becomes so confusing, right? So what I have tried, that this is literally like a story, right? Yesterday someone was telling, right, about the story part. Yes, this would be exactly like a story, all the way from the step 1 till the end, and then you will see that literally, like those baratis, right? Whenever there is marriage, so then those baratis come, and then how this progesterone takes over, how and let's say if you are attending any of the marriage, so then there would be a welcome drink, right? So there will be a welcome drink, and then they say those baratis, because they are very hungry, so then you have to supply more food, and then there would be a gala dinner, right? Exactly all those things would happen, and one by one all those layers would be formed, right? uh what exactly, Zaid, what exactly you mean by that? That इस week की notes email पर नहीं दिख रही है? Well, I haven't sent any notes on email. That's right. Right. So this is good, right? I was just watching some of the comments that you guys are also helping each other. Yeah, exactly. Zaid, he's right. There is a link in the description, and when you go to that link, it will take you to a folder. It is like a folder on the cloud, right? And that folder will be the place where all the notes will be lying, right? So there should not be any problem. Okay. So let's start with the entire process. So here it is, the first fact, that the sperm is the smallest cell and the egg technically called as the oocyte is the largest, right? The difference of the size is about 30 times. This is 30 times bigger. But when they both meet, right, there is formation of what's called as the zygote, right? Few words I'll keep on repeating so that you remember them properly. As you must have expected, yes, I'll be asking several questions from today's session, right? Because we'll be taking this journey all the way till 3 weeks. That is the point where the trilamellar disc is formed. Don't worry about the question bank, right? Don't worry about the question bank, right? Whatever we discuss from Monday to Friday, right? That is what will be included into the say those tests. And honestly speaking, sometimes what really happens? No. I'm coming to that. Sometimes what happens that what you said is practice, right? Actually, it is not practice. Let's say if I give you 200 questions, right? If I give you 200 questions, that okay, these are the questions which you have to say practice. Actually, you'll do it 3 times. At the fourth attempt, you don't have to even think. Because whether you are knowing the reason or not, it will definitely be, you'll be clicking the correct answer because it will come into your reflex action. It goes into the short-term memory, right? So actually it is not reading. It is not studying. It is not even understanding the topic. You pick up any of the complex topic, right? You do those MCQs 3, 4, 5 times, but done, khatam. Then you keep on boasting that yes, I scored 90, 95, this, that. But when it is asked, okay, can you repeat the entire process? Gone. That will be a big trouble. So that's why I'm insisting on only one thing. Don't even worry about, don't even struggle on that. Okay, let's find some questions from here. Don't do that. Don't do that because we are not comparing your result with anyone. But yes, go through these notes very well and then see how you really enjoy that exam, right? Because questions would be asked in such a way that you'll really relish it, you'll really enjoy it. And even if it is, let's say, I would say out of 40, what if I answer 20 questions incorrect? Doesn't matter. When you'll discuss, right? When we'll discuss, at that point you'll come to know that why it went wrong. It's like a detective story, right? It's like a detective story. When we read any of the detective novel or movie, right, we guess that okay, she is the murderer, right? Because, and in case if it turns right, so then we keep on boasting, right? Because I am born smart, yeah, such things, so I solve it so fast, no problems. And in case if you are wrong, so then you hit yourself कि यार इतना बुद्धू इतना भी पता नहीं चला, right? I couldn't even guess this much. But in both the cases, as it is involved in what is called as the active learning, you'll always remember it, right? So I don't want any of you to go for that passive learning. Just answer is B, C, A, this. No. Right? It is completely fine if someone tells that, okay, I answered all 40 incorrect. No problem. I'll appreciate him. Because then in the second test, he'll be doing wonders. So same thing is over here. Don't worry, study the topic and you'll be very happy, right? Just let one say this exam go and then you'll really like it, right? Because it is like, that's why I always say it is like an honest exam. Okay, right? No exam would be like exactly like this. Say for example, this would be one page, right? There would be a question and then A, B, C, D, right? And you have to just select the option, and on in your notebook you'll just write, just keep it ready like 1, 2, 3, 4, so that you don't waste time at that point of time, right? Just one all the way up to 40, right? And you can do one more thing, you can keep some space between 1 and 2, so that when we go for discussion, in case if you feel like that yes, this is the point which is important, so you can just jot down that point in between. And if you're right, so then great. If there is anything incorrect, no problem, in that space you can even write it down, right? So that is like a very simple thing, but very enjoyable. You'll like it. Okay, all right. So Great. Yeah, so that's how the exam would be conducted. Okay, so let's start with, once again, right? This is just a very brief thing what I'm writing. Sperm and the oocyte, secondary oocyte, right? 30 times bigger, and when they unite, right, there would be formation of zygote. And yes, we'll be watching all the steps, right? Now, it's like a mission, right? It's like a mission, and 200 plus million brave sperms, right? They take part, right? And they know it very well that this journey is not going to be easy, right? Definitely, this journey is going to be very difficult. The reason behind it is that this, say, vaginal environment is acidic. So many of the sperms, right, they will be sacrificed, they will be destroyed into that acidic environment. At the same time, the path all the way from vagina to reach to that ovum, that is so cryptic, so many of them would be lost in the way, right? In fact, WBC of female, they will also be fighting with these sperms because these are foreign bodies, right? So in that war, many of them will be sacrificed. So that's like 200 plus million soldiers, right? They will start marching and then see what really happens. The important part is that all of them, they are into that seminal fluid, and this fluid is in fact alkaline, right? So right from the beginning, right, the all the inventories are ready, and they say that because this is alkaline, so we'll try to neutralize, right? We'll try to neutralize that environment so that some of the sperms, they survive, right? Okay. See, now we'll be going for those MBBS books for MBBS, right? Just give me some time. We'll definitely be going for it. For next just 1 hour, right? Don't write anything. Don't write anything. This entire process, right? It is so interesting. Trust me, it is so interesting, and step by step, right? You will be wondering how, and those who are seniors, so they already know it. But take it like that what really happens, then that one cell, then that it multiplies, then all the way it goes to, say, trilaminar disc, and then formation of all those structures. So I'll be taking you to that point from where, when it will come to, say, development of heart, we'll pick from that point. Okay, these are the 3 discs, and this is what really happens, and that's how the heart is formed. When we'll be talking about brain, from that point only we'll lift that, okay, this is how the brain will form, right? Okay, so let's pick up now 200 plus million sperms, right? So they are into that seminal fluid, and here it is that that environment is alkaline. So this is the preparation. Right from the beginning, they prepare that, yes, this is going to be a difficult journey, right? And it is acidic, so let's try to neutralize it, right? One more thing, that all these sperms, they know it very well, that see, this is how the sperm is, right? That there is a like a head, and then there would be a wiggling tail. So they'll say that it is this head end. This head end should be well protected, because as we'll see that this head end is consisting of some very important structures, right, which will be utilized at the end. So that's why give them what's called as the protective glycoprotein, right? Protective glycoprotein. So this is what will protect them. And yeah, trust me, Bharat concept. Yes, seriously. Okay. I think You are right. Means I'll not see the chat much now, right? Because honestly, I'm distracting. And what really happens that there is a screen on this side. So even if I'm watching towards you, but the moment someone writes na, in the peripheral vision, it shifts. So nazar goes over there, right? It goes over there. Okay, no problem. But yes, for now sometime I'm telling you, I'm sorry. Even if you write something, I won't see, and I won't even reply, right, for some time, because let this entire process flow, then and then you'll enjoy. Otherwise, with interruptions, seriously maja nahi aayega, right? Okay. So here it is now. Our 200 million soldiers, they are ready into the seminal fluid. This is alkaline, right? And they know that they'll be entering into that acidic environment. And these sperms, they will be having that tough helmet, right? Protective glycoprotein. So that is there. Now, with that intimate moment, the journey starts, right? And say that's the uterus, right? That's the uterus, and these are the fallopian tubes, right? That's the fimbrial end, right? That's the fimbrial end, and here is the ovary that is suspended. This is the ovary that is suspended, and one by one on both the sides, right? Step by step, right? Every month, one ovum would be released. So let's say this month it is on this side. So our entire process now will be starting over here. So all those sperms, they entered, right? This is acidic environment. So initially in that process, many of them will be lost. Now they will start moving, right? Many of the sperms, they will be going on the wrong track because there is no ovum over here, but well, can't help. And some of them, they will start traveling on this side, and somehow they reach over here, right? They reach over here. During this entire journey from vagina to all the way to say uterus to fallopian tubes, and when they reach over here, right? Very sorry to say, but out of those 200 plus million soldiers, right? Many of them, they lost their way. Right? They lost their way. So lost the way, and they went on to the other side. So now nothing can be done. They can't come back. Gone. The inside is the mucus environment. So many of them will be entangled over there, right? They are unable to swim, right? They are not strong enough, so they just got stopped over there. And some were brave, right? But they lost their life. So they lost their life in that fight with WBC. See, that's why in one of my session, it was like a motivation session, I was telling that why any one of you should ever be going in depression, because say you won the first fight of your life, right? And in that fight, there were more than 200 plus million competitors, and you are the best. And that's the reason that it's like you are the best. There is no comparison, right? Because what are the chances that out of 200 plus million, only one would be successful, and that successful guy was you, right? So why to worry? As such, right from the beginning, you are the best, right? Over here, see what happens. It's the great fight, but in that process, so now we have got about only 1,000 finalists, right? Only 1,000 finalists are there. Now, out of these 1,000 finalists, they know that now almost they have reached to the end, right? So now there is no need to keep that glycoprotein intact, because the head itself is consisting of something which would be needed at now to the next point, right? So they will start rubbing their head, right? So these 100 finalists, they will rub their head against the wall, right? against the wall, because that inventory is over here, right, which will be used. Because see, still difficulty is not over, right? Still there are so many problems. So they will rub their head against the walls. And the purpose is just one, that glycoprotein, right, protective glycoprotein is not needed. So let's remove it. So that protective glycoprotein, glyco glycoprotein is removed. So I'll write it like this. So that glycoprotein is removed, right? And now this head is ready. So this head will immediately release, right? Because it is not only the glycoprotein, but even the plasma membrane is removed, right? So I'll just write it like this, plasma membrane. So even that is removed. So basically, it is like just cutting down the head. Right? That's why its name is also capacitation. Capacitation. So now this head will do, it will secrete, right? Let's change the color. Right? It will secrete an enzyme, hyaluronidase. Whenever the A's comes, so that means it's an enzyme, right? It's an enzyme. Why do we need enzyme? We need enzyme because now these 100 participants, 100 finalists, they are about to encounter the final, that is that ovum, right? They are about to encounter that. So first thing, that egg, that is not going to say give the easy entry, right? This egg is telling that, well, that things are not that cheap, right? You have to really work hard. See what happens even at that molecular level, right? So what this egg has done, it will say that, see, this is hyaluronic acid. That's the first barrier. These are like, say, in games there are like level 1, level 2, level 3, same way over here, right? Once this hyaluronic acid barrier is broken, so then there will be corona radiata. Once that level is cleared, then there will be zona pellucida. Once that is clear, so then this egg will say, okay, right, you are good. And then these finalists, right, whosoever will survive, they have to come into the role of Spider-Man, right? So they'll become Spider-Man. And then out of those Spider-Man, one Spider-Man will be very smart, right? He'll take the entry, and then what happens? Let's see. Okay, so here it is, this hyaluronic acid. So this enzyme, right, it is telling that I was knowing it very well that this is going to happen. So here is now my answer. Hyaluronic acid, right, it will be neutralized by hyaluronidase enzyme, right? Hyaluronidase. So now first level cleared. Hyaluronic acid is broken, right? It is broken. So level 1 or the layer 1 down, right? Let's move on to the layer 2. Now comes the second layer. Obviously, in games, as it happens that level 1 is relatively easy, right? Level 2 is bit tough. Level 3 is still tough, right? Same thing, right? There is nothing new in this. Same thing would happen. So now this egg would say that, see, I can't give entry so easily, right? I am not that easy. So the second layer. Now this layer is like follicular layer. So there will be so many cells, right? So even this egg is also giving a tough fight. So here it is, right? Comes the second layer. Now I'll draw it like this, right? This is a second layer. So that hyaluronic acid layer is gone. So that I'm not drawing. Okay. This layer is corona radiata, right? It is what is called as the corona radiata. And you can say these are, there will be follicular cells, right? There would be follicular cells. So now our fighters, they reach to this level. And still there will be one more layer, right? Even a third layer. So it happens that when they reach it to this point, right? out of 1,000, only half of them they are successful, right? Only half of them. So about 50%. So only 500 they reach to this point. And then say this would be the layer 2, and then let's write layer 3 also, and then we'll be discussing about all of them. This layer 3, right, once this is negotiated, then there would be zona pellucida, right? Zona pellucida. So this would be the third layer. See, the names are also getting tougher, right? Hyaluronic acid was a sensible name. Corona radiata, easy to remember. Zona pellucida is a cryptic name, right? Slightly different. But because it is level 3. And then inside the zona pellucida also there is something, right? There is still more interesting. We'll just write down it as ZP3. What ZP3 is, we come to that. Okay, so let's see now what really happens at this third layer, right? Second layer. Why so many people? See, now what I have to do is, because if this thing would continue, so then I have to, hyaluronic acid is also called, that's okay, that's okay. See, I have to stop the chat. I think, should I stop the chat? I really trust you guys. But still, it is like if you keep on writing in some other language, this, that, don't do that. And because say I trust you guys so much that I feel that once said, you'll follow it properly. Now there is no point in writing any of the other language. Why to do that? Don't do it. So many people, right, when they are studying, I think I really don't wish to do it, but how many times I have to say that yes, there is test tomorrow, right? So please don't type. Now don't type. I don't feel like stopping the chat, but still, if yeah, because now almost everyone is telling, so yes, I'm sorry, but I have to stop the chat. Right? I really don't like it, but I have to. Okay, if people are not typing, so then I don't do it. Great, chalo, right? I'm still, the menu is open, right? Really stop? Okay, sure. I have stopped the chat. Okay. I never wanted to do that, seriously, because it hurts me, right? But anyway, chalo. But there was so much of distraction, so that's why there was no way. So here it is, right? I'll turn the chat on after some time. So here is layer 1, layer 2, and layer 3, right? These are the 3 layers, layer 1, layer 2, layer 3. Layer 1 was like hyaluronic acid, right? That was the enzyme. And then second one was corona radiata, that is layer 2. And the third one is zona pellucida. And in that we talked about that there would be something like ZP3, right? One special layer. It is like a bonus round, right? So this ZP3 is like a bonus round, right? That's the point when our fighters, they'll become the Spider-Man, right? So here it is, that head is gone, right? Hyaluronic acid has been released, right? And this hyaluronic acid, it as it released, so now our fighters, they are on level 2. At level 2, what happens at level 2, right? So level 2 was that corona radiata, right? That corona radiata. Now this is a tough layer radiata. So we need to cross this. So in order to cross this thing, right, there is release of acrocin, right? Acrocin. Acrocin is released. That is by the sperm. Acrocin is released. So what acrocin is, it is like a hydrolytic enzyme, right? Again, it is an enzyme. Yes, everywhere there would be enzymes. So it is like hydrolytic enzyme. So this hydrolytic enzyme will act and it will drill a hole, right? It is like a drill work. It will drill a hole and it will cross corona radiata, at the same time zona pellucida. Now zona pellucida is reached. Now interestingly, that this is the layer, right? That's the zona pellucida. So if I draw it like this zona pellucida, right? Just underneath the zona pellucida, just underneath, there is one more layer, right? This is underneath. And that is, there is, it is what is called as the ZP3. That is zona pellucida sperm anchoring protein number 3, right? So it means that all those difficulties, right, all those difficulties which were over here, right, but finally there was a good support over here. So once those fighters, they reach to this point, right, they drill a hole into zona pellucida, so then they know that yes, these are the anchor points. So at that point they become like Spider-Man, right? So they will start assembling those protein, and these are the actin proteins, right? So what I'm telling is that these are the actin proteins, right? Actin proteins, and these sperms, like a Spider-Man, right, they will throw this anchor protein, actin protein, and those proteins, they will be attached to ZP3, because this is ZP3. ZP3 is that layer which is zona pellucida sperm anchoring layer. So that layer is a supportive layer, right? That's why we said that it would be like a bonus, right? So thus that actin proteins, they get linked. Once they are linked, now things are easy, right? Because now it happens that literally due to that actin protein, this sperm would be pulled in, because now it is like that ovum has accepted that sperm, right? Is like enough of, say, exercise, right? No worries, can come in. Now here it is. When they come, right, the acceptance level is so good that immediately, right, the plasma membrane, right, immediately now the plasma membrane of both, right, is of sperm and the ovum, right, they both fuse, right? They say now it is all good, and this is what is called as the sperm binding. Now the sperm is bound, right? Sperm binding. Now meantime, inside environment, right, there is something very interesting going on. So here is the entire battle was going like this, right? This was ovum, and here is all those sperms, right? They were fighting, right? They were struggling, and they were, some died at the level 1, some died at level 2, right? They were entering, and one guy, right? He reached, right? He reached, and well, this is the selected candidate. Now, inside environment, just over here, inside ZP3, right? This entire space. So there are few granules, right? There are few granules. Those granules, they are not used. They were not getting used till this point. And all these granules, they are loaded with enzyme, right? They are loaded with peroxidase. Peroxidase. They are loaded with peroxidase. So this peroxidase, as soon as that one sperm, right, he penetrates, right, and this ZP3 helps, immediately it happens like that. It is like a message, right, that okay, candidate has been selected, and in that process, that candidate, right, that is that sperm, as soon as he is selected, right, there will be lots of calcium release, right? It is an indication that okay, candidate has been selected, and this is what is called as the cortical reaction, right? Cortical reaction. Now this cortical reaction will send a message to every granule that okay, right, he has arrived. Okay, so all of you burst, right? Thak, right? All of them, they burst, right? And then in the process, there would be release of this peroxidase, right? So peroxidase is released. So that is what is called as the cortical reaction. So now this peroxidase is released. Now, why was it necessary to do like this, right? What it was telling, right? It is like when the peroxidase is released. So this peroxidase, along with those glycosamines, right? Those glycosamine, then there is water, right? Then there is calcium, right? All of them, they'll say, okay, make a layer. So that layer is what is called as the hyaline membrane is created, right? What hyaline membrane is? Hyaline membrane is that, okay, interview is over, candidate is selected, so rest of you get lost, right? That's the actual meaning, right? So it happens like this. So this was the layer, let's say this was ZP3, right? Anchoring. Everyone was trying to anchor, right? But the moment one entered, right, immediately this layer is now sealed, right? It is like that's hyaline membrane. And only this one gentleman, right, he was allowed to enter in. And rest of them, they are said that, okay, interview is over, all of you get lost, right? Done. Game over. Sometimes if this process is bit delayed, right, for any of the reason, this is a chemical reaction. So sometimes, say, though that calcium was bit less and it took some less time, right? It took some more time. If there is any delay for any of the reason, then obviously two candidates, they have entered, right? Two candidates entered. When two candidates entered, that's okay. That's what would lead to twins, right? So over here, normally it won't happen, right? Because this entire story is very meticulous. Now, meantime, there is story inside. Story inside. Inside, what is happening, right? Inside this ovum, there was that meiosis 2, it occurs, right? So which leads to that division, and in that one is the small body, and second is that entire daughter cell. This is the daughter cell which is so vital, and this daughter cell, this becomes what is called as the polar body, right? So during division, this polar body, it goes into self-destruction, apoptosis, it goes away, but it is this daughter cell, this is what is keeping every piece of information. So there are 23 chromosomes in this, right? It is this. So actually now this daughter cell is ready, right? It is what is called as the mature female egg, right? So during the entire process, right, in the stage of, say, what exactly is this menstrual cycle? Just nothing. Every month, one of the ovary, it releases that ovum, and then immediately the entire system feels that yes, this ovum is going to get fertilized, and let's prepare the uterus, right? So the progesterone takes a lead role, right? Progesterone will place a call and will tell all the arteries, and will send a message in the blood. That message will reach to the uterine arteries, and they will be told that yes, anytime the fertilized egg would come, right? So you do one thing, you just make the entire area rich, lots of blood, lots of vessels. And then if nothing happens, so then after 10-12 days, it will say, okay, fine, break the entire structure, right? That is what is nothing but the bleeding phase, right? And then once again, the next cycle starts. So that when we'll be studying the entire menstrual cycle, right, we'll study the follicular phase and luteal phase and everything. But over here, the what is happening is that the fertilization has occurred. So we are now, actually the entire embryology is into three parts, right? This is the cycle, right? Menstrual cycle. Once the fertilization occurs, right, this is what we are studying today, right? And then finally, from this point onwards, all those organs or tissue development, right, which is very specific. But this area is that area where there can be lots of confusion. So that's why we have picked up this area. Okay. So now that mature female egg, right, will now call this as female pronucleus. Pronucleus. This means that everything said and done, this female pronucleus is ready with its 23 chromosomes share, right? Now over there, when we'll see the process of sperm formation, again all those divisions, etc. But finally, what happens when that one individual sperm is also carrying 23 chromosomes, right? So it is carrying that thing in and it reaches over here. Now when it has reached, when it has entered into the environment, right, now it is into the system, right? It has passed through all the layers. Now over here, head is lost. So now it gives the final shot. It has got just a tail. There is mitochondria, right? Mitochondria is like those batteries, right? Powerhouse, right? It will say mitochondria, okay, even if there is 5% battery, right, let's swim this final dash. So this is the final run, right? And that's what is called as the male pronucleus, right? So it means pronucleus is what? Pronucleus is that final component which is, after all the struggles, it is the final output, right? Finally, even this tail and mitochondria is also lost, and the output is that male pronucleus and the female pronucleus, which was already there, but she also divided and she is also ready, and this reached. So here is now both of them. Now when actually this thing occurs, right, they both are very happy, right? So literally there would be a sort of a dance ceremony, right? It is like a dance, right? And then they both release their genetic material, right? They'll say, okay, this is my genetic material and show me your genetic material, right? So all those chromosomes, etc. And then there is matching, right? That is what is called as the complementary pairs are made, right? Complementary pairs, they are made, right? Then there'll be on both the poles, right, there'll be formation of those chromatids, etc. But the point is, it is ready now, it is done. And then both of them combine, right? They both of them combine, and then comes the first thing, right? This is what is now called as the diploid. Diploid means double. This is like 23 into 2, right? 23 from both the sides, from the mother, from the father, right? From both the sides. So now we have got 23 pairs ready, everything fine, done. And this process, this is what is called as the zygote. Zygote, right? And say during dance, they sing a song also. So this is what is called as the singami, right? Actually, this is not the spelling, right? The spelling is Y, singami. Okay, and let me write it bit properly. So that's to remember that they, with the dance, they also sang a song. So this is singami. Singami means single cell. So this is a single cell. Clear words: male pronucleus, female pronucleus. Male pronucleus, all the way from all the struggle from 200 plus, 200 million plus, right? Those soldiers, 1,000 finalists, 500 finalists, crossing those layers, right? Releasing hyaluronidase, releasing actinolase, and then drilling it, and then finally anchoring to the last layer, and then releasing all those peroxidase, right? So that no one else can enter, and finally successful. And then there was a dance in which both of them, they released their genetic material, and finally both of them combined their plasma membrane, and they lead to formation of one single cell. This one single cell syngamy is called, and this entire thing is called as the zygote. From this point onwards, right, when this thing occurs, from this point, and this thing is, it occurs within 4 hours, right? Within 4 hours, these things have happened. Now meantime, see, the journey is not that simple, right? This is the fallopian tube. This is the fallopian tube. And as those fighters, they were traveling on this direction, and then that ovum was released over here, and somewhere over here the fertilization occurred. The moment fertilization occurs, right, so then this zygote tells that this is not the way to work, right? I need to go back to the uterus, right? Because that is the place where it is necessary to embed into the walls, and then that would be the place where things will become bigger and bigger, and the baby would be born, right? That's the place. So how this entire thing starts? So over here it's okay, right? I'm just writing Z. Zygote is formed, but then this zygote starts his journey all the way from this fallopian tubes, right? Via these fallopian tubes. It is important because if this journey is not successful, right, over here there'll be all those cilias, right? So they'll keep on pushing, come on, go, right? It is like not only this zygote is moving, but at the same time the process, they are multiplying also. So many things are happening. In case if this journey is not completed in time, so then it would happen like this, it would be embedded here only somewhere. This is bad, because fallopian tube pregnancy, that is what is called as the ectopic pregnancy. No scope, because later on you will find that there would be so many Baratis, and this small place is not sufficient to accommodate all of them, right? So it's like a bad end. So let's not think about it, right? Right now things are going good. So this zygote, right, he is good, right? He or she, right? We can't say at this point of time, but say it will start. So this zygote, right, which was one cell, that's a process what's called as the cleavage, right, divides. So now two cells, then from 2 to 4 cells, from 4 to 8 cells, from 8 to 16 cells. They divide and divide and divide, and now a new name is given. It is called as the morula, right? Morula. When this much is reached, it is the time when it will say, okay, morula is like this, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Just a ball. Now these cells, they say that enough, right? We need to improve. And what really happens is these cells, remember this word, they differentiate. Differentiate means they change their characteristic, they change their structure, right? Everything is changed. So out of all these cells, right, now they are dividing. So they say that okay, few cells you take the cover, you just take care of the boundary, and there are few selected cells, they'll say that will go for the process of formation of that baby, right? So here is what really happens, and now we'll start giving the names. Okay, let's make this thing white, right? That would make much better. Right, so few cells, they will take the border. Now they are dividing, right? They have divided. Now there are large number of cells, so few cells, right? So they form the boundary. So we'll give them some name, right? Now these cells, those who are forming the boundary, will give them the name trophoblast, right? Trophoblast. You know this blast, whenever we said blast means formation, clast means to cut off, and sites means to maintain. Over here, everything would be like blast only, because they said that we just don't have to maintain, we have to keep on making, right? So many things. So these are called as the trophoblast. They'll say that, okay, will become epithelial cells, right? Epithelial cells. So will protect the inside environment. Done. Those selected special cells, right? They'll say that we shall call ourselves as embryoblast, right? It is this embryoblast who is going to become a baby, right? All the way, right? Right now it is so simple, but see what really they do. So that is also embryoblast. With some space, yes, there is some space also created, right? Because these cells, they differentiate, and then those cells, they come at one point. Now this is what gives one more advantage, and it tells that it gives the pole, right? That it is this side, because otherwise if everything is round, then how will you say which is head end, which is tail end, etc.? Nothing, right? So here it says that wherever these embryoblasts, they will be accumulated, we'll call them embryonic pole, right? So this is now called as the embryonic pole, right? Embryonic side. So this entire thing, and this space, right, it is called as the blastocele. This space. So blastocele, trophoblast, embryoblast, collectively we need to give some name, because this is now a new structure, right? So let's give it a name, and that name is blasto, because everything is blast, right? So it is blasto, and cyst means fluid filled cavity. So whenever, right, later on also you'll come across this word, very common word, it is like fluid filled cavity, fluid filled cavity. So it is now called as the blastocyst. Now this blastocyst, it is this point when blastocyst will start making some changes, because now this blastocyst is telling that I think uterus has arrived, right? So now this is the high time to take care of it, right? Because it tells that ab kab tak bhatkenge, right? And almost it's like without eating anything, 4 days have passed. This is like 4 days, right? And this is all rolling and tumbling and then dividing and then somehow surviving, right? So 4 days without food, without water, nothing, right? Still they divided. So now it is the time, and this is where in these 4 days it has arrived in uterus. So uterus arrived, right? It will say that yes, it has come. Okay, now this entire process was happening. How was it happening? Right? Into the ovum, this blastocyst has formed, correct? Right? Into that ovum, right? this is what really happened, right? Because this was the entire environment. This was the entire environment, and then male and female pronucleus, they merged, right? Then one cell, then divide, and then finally the blastocyst was created, right? The entire environment is over here. So it will say, okay, now let's go out of this ovum, right? This surrounding is now not for us, right? It is this piece which is so important, right? It has to go and embed into the uterus. So I'll just mark it that this is blastocyst. We are on day 4, right? So now this blastocyst, it reaches, right? It is the point when it comes out and it reaches to this uterine wall. So just think it like over here, right? And like this, and it is over here. So what it does, if we zoom this portion, right? If we zoom this, here is that blastocyst, right? And this is the uterine wall. So over here, making it bigger, right? And now onwards, I'll just draw it like this. So this is, you have to keep on remembering the name. I'll also keep on telling, right? So this was trophoblast, correct? It was trophoblast. And then we were having these cells and they were like embryoblast, right? Embryoblast. But this much you remember. And yes, that was blastocele, right? This was blastocele. And here is the wall, right? Here is the wall. This is the wall of uterus. So in this uterine wall, it tells that we need to penetrate into this, because this side is what is called as the decidua. Decidua is the uterine wall, and that is having so much of rich blood supply, so many vessels, right? And the good environment to settle in. So it tells that we need to enter, right? Now things will not be that easy once again. So again, right? Let's go back to that nature of Spider-Man, right? So it tells that, okay, special proteins out, right? So these are all selected proteins, right? And they are called as L-selectin, right? L-selectin protein, right? These are L-selectin protein, right? So I thought that let's write it like this, right? L, right? L, right? These are all L-selectin protein. And these selectin proteins, they have got affinity for carbs, carbohydrates, right? Carbohydrates. And it actually makes sense also, right? Because in marathon, when you run, right, you are given carbs, right? You are given carbohydrates, because say you have run say 32 kilometer, like it is difficult, mare se, because now from this point onwards, so running is difficult. So then the carbs are given, and once again you are active, right? You can run. So over here it tells that chalo, these are the proteins, right, which have got affinity for those carbohydrates. And then there are carbohydrate receptors, right? These are like receptors. So immediately they would make a contact. Right? So connected. Now they are just connected, right? Nothing much. Now comes when they are connected, right? This blastocyst, right? This blastocyst said, now is the time that now I need to show some style, because now it is point to go inside, right? So here it is, right? That is what? Trophoblast, right? And in that there was embryoblast, right? And somehow reached to this uterine wall, and over here there would be differentiation of cells, right? Differentiation of cells. Those trophoblasts, which were so Siddha Sada, right? Those trophoblasts, they were all very simple cells, right? Simple epithelial cells. And suddenly they become so stylish, right? And right, there should be some sensation, so they become sensational trophoblast. And don't write it in exam, sensational trophoblast, because the real name is syncytial trophoblast, right? Syncytial trophoblast. But I always remember it like sensational trophoblast because it looks good, right? So this is like syncytial trophoblast, and they are like tick, 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 right? They are like this, right? Like this. So how are they different? How are they different? They say that, well, others are single nucleated, we are multinucleated, right? Multinucleated. So these sensational, right, syncytial trophoblast, right, they will try to enter, right, enter into the decidua, enter into the uterine wall. This is uterine wall. This is uterine wall, right? Now from this point, there comes a phone call, right? That phone call comes from corpus luteum. What was corpus luteum? Corpus luteum was that blank tissue which is left out after the ovum was released, right? Ovum was released. So this is like that crumpled structure, right, which said कि yes, that I have released the ovum, and if this is fertilized, then it should be taken proper care into the uterus. So this corpus luteum, it secretes progesterone, right? That is what is called as the secretory phase. So secretory phase, right, it releases progesterone. Now this progesterone is super powerful, right? The management skills of progesterone are so good because progesterone tells that every month I was doing this work, right? So every month in anticipation that yes, I have to welcome this Bharat, right? So I was working. So that was in anticipation. This time also it will immediately place a call, right? That call is like progesterone would be released into the bloodstream, right? And when it is released into the bloodstream, it will reach to the uterus also, right? So its function is to prepare the uterus. In case if there is no fertilization, as we talked about, that after 10-14 days, this will become inactive and all the prepared structures, they are broken, and that is what is called as the bleeding phase, right? So over here, this when it reaches to the uterus, right, in the blood vessels and the arteries, right? So uterine artery, they become large and coiled, right? So this entire thing occurs by day 7. So in day 7, we have reached to that point that now that blastocyst has reached into the uterine wall, right? It has not yet fully penetrated, but yes, it has already started penetrating, and at the same time progesterone has been released, and here are all the blood vessels which will be over here. So those blood vessels, right, they are just increasing in its number, right? Just increasing in its number. Still they are not doing anything. The real journey starts, the real work starts into the second week, right? Now the second week has started. So in second week, we are on day 8, right? On the 8th day. See, this is the uterine wall, right? That is also not easy to break But well, because of thanks to that sensational trophoblast, right, what it really does, that it is over here, right, and this is the trophoblast, and this is like that, right, exactly, embryoblast, right, embryoblast, and then these were all those syncytiotrophoblast, right? And as we said that these syncytiotrophoblast are multinucleated, multinucleated. So when they are, right, these are syncytiotrophoblast. So these trophoblasts, they said that if you guys are getting a good name, so then give some name to us also. So they'll say, okay, chalo, you are like simple cells now. So you, now onwards, you'll be called as the cytotrophoblast. Otherwise, trophoblast and cytotrophoblast, they are almost the same, but cytotrophoblast is those bichare trophoblast who are unable to get that multi-nucleus, right? And they were not allowed to take part into this sensational thing. And so these are like Siddha Sada, single nucleated, right? Single nucleated. So they are now called as the cytotrophoblast. And these are sensational multinucleated. Right? This entire environment, this one, right? This is inside the uterine wall and it is called as the decidua. Right? It is called as decidua. So obviously this point, right? It is the base from where things are entering. So this is what is called as the decidua basalis. Decidua basalis. Right? This is day 8. Day 8 is complete, right? And this much work has been done. Now starts the entry. Now this entry is going to take time, right? So almost for next 3 days, it will be a day of struggle. So day 9, day 10, day 11, right? Only one thing is getting done. And he does it successfully, right? Because the moment I'll draw, you'll come to know that how successful it is. This is the uterine wall. And now the situation is like this, right? Like this. This is syncytiotrophoblast, right? And that's our cytotrophoblast. Cytotrophoblast. And inside is having that embryoblast, right? This embryoblast is still waiting, right? Still waiting. And still it is telling that, see, we have reached to this point and still we have not consumed anything. Kuch khaya piya nahi, right? So now it comes like on the day 12th, right? This is like day 12. That progesterone tells that what you guys are doing, right? They have come all the way and still you have not even given them any food. So immediately, because of the call of progesterone, right, there is a decidual reaction, right? There is a decidual reaction. And in that decidual reaction, those cells which were inside, right, the cells, right, which were, because this is decidua, right, over there, there will be so many cells. Those cells, they suddenly becomes waiter, right? So now they are like sugar rich fatty fluid, right? These are the cells where that thing, these are the cells of decidua, right? These are the cells of, I'm writing D, decidua. They become sugar rich fatty liquid or fatty fluid, right? So that becomes like a welcome drink. So this syncytiotrophoblast will say in style, that's okay, that no problem, right? So just give me those things and I'll supply it to our embryo Mahashay, right? Who is developing. So he is in need of some energy. So this is like the initial energy, right? You arrange for more, but meantime, this is a kam chalao thing, right? So that mithai that is sent, and these are nothing but cells of decidua. Meantime, progesterone is calling again that, see, he will be increasing in its size. So why only few of the decidual cells they are in wait in this process, right? Deploy more. So they'll say, okay, if progesterone is telling, so more cells, more cells, more cells, right? All of them, they will, right? Dord ke, yes, yes, you have this food, right? And that is all thanks to, say, thanks to progesterone, right? See how enthusiastic the progesterone is, right? So due to this, the best is he sustains, right? He sustains, because since so many days no one gave any food or anything, so this sustains, and that is what is called as the early survival, right? It sustains the early survival. So that's sort of welcome drink, but this welcome drink is not something, it's like a full-fledged food, है ना? So they'll say that initially to it's okay, but now we need some more food. So that way, slowly and gradually, the day 14 has arrived. Right? Day 14 has arrived. So in day 14, this is the situation, that was the wall, and that's our cytotrophoblast, correct? So I'm just writing this white line is, if I'm writing CY, cytotrophoblast. Inside that, there was still lying, right, the future baby, right, and that is embryo trophoblast. And then those sensational trophoblasts, sensitiotrophoblasts, right? They are like everywhere, right? They developed even on other sides also, right? And they are just consuming whatsoever is offered them into the form of that welcome drink, right? They are just sustaining. But these say sensational or sensitiotrophoblasts, they'll say, okay, this is not something which is going to last longer. So they start developing long projections, and they are called as the primary villi. They say that very soon large amount of food is going to come, so let me create the channels, right? So it starts creating all those projections, and these projections, they will make its connectivity more powerful. So these villi, they expand, right? They expand. These primary villi, they expand. Now when they expand, so they'll say to all those decidual cells कि चलो make the space, खाली करो जगह, right? So that means those decidual cells, they start clearing out. They start clearing out. When they clear out, because they are getting eaten, right? So there would be formation of lacunae, space, right? There would be formation of lacuna. So that is what is called as the lacunae, right? Just for orientation, this is the situation, right? This is the situation, right? And that's the uterine wall. And all these lacuna, they are getting formed surrounding over here, right? Progesterone also called up that all those vessels, what were you doing, right? Progesterone said vessels, that is arteries and veins, dilate, right? Be rich. So then all of them, right, they will be increasing in its number and the coils, right? So they'll become larger and larger. Meantime, these lacuna, they are also getting together, right? They are joining together. So now it is like a space is created, right? Now we can't just say lacuna, it is like a real space is created. It is like a large pool is created, right? Large pool is created, and that's the point where these blood vessels, they break into it, right? It means these pools, now they are filled up. They are filled up with blood. Now these pools, they are surrounding this hungry embryoblast, right? And those primary villi would say, okay, now no problem, right? So I am now directly connected. So that is how things really happen. Still going deep, right? these primary villi, right? So you got the idea. I'm just turning the chat on just to know that till this point is everything okay, right? And I'll keep it on if there is no issue. I'm enabling it. Done. Now you should be able to type. Yeah. Right. In case if there is any query, just let me know. In case if there is no query, don't write no query, everything is fine, right? I'll understand that things are good. And we'll be going for a very good high-speed revision also at the end of our today's session. So even if there is some query, right, I'll definitely be solving it. Okay, so here it is. The situation should be like this somehow. Last page, 1 second. Sure. That's what I was about to do. That this is the situation because it was drawn in part. Now I'm drawing it in full. So this is the uterine wall, right? Uterine wall, and that's the cytotrophoblast, right? So cytotrophoblast. That was the embryoblast. Okay, and then there was our stylish syncytiotrophoblast, right? And then there was a pool, right? There was a pool. Just a minute. Too dark. Okay. So there would, because of that formation of that pool, this is the situation. This is the situation, right? Those blood vessels, they broke into this large pool. So this is the final situation, that there is blood, right? There are all those tortuous, right, uterine vessels, they are there, right? And there is syncytiotrophoblast, there is cytotrophoblast, and there is embryoblast. Okay. Corpus luteum is not knowing that ovum is fertilized. He is just making a guess that every, right, work hard for 10-12 days, and in case if there is no other, say, reply, right, it will stop working and it will break down, right? So it is doing its duty, right? What is the use of fluid present in blastocele? Well, that is the fluid. Actually, that is the fluid which keeps on going, giving some chota nutrition so that it survives, right, during this entire journey. So it is like a small sandwich, right? Primary villi is formed from cytoblast. Yeah, that's right. That's right. In stage 3, who releases calcium? Well, there are so many mechanisms of release of calcium, but when we say that, you can say that they are like gated systems, right? In fact, you will be understanding it very nicely when we'll be talking about neurotransmitter, because it will become a very long answer that what really happens when the chemical leads to release of increase or decrease of the calcium or any of the ions. Say this entire journey is of second week, right? Second week. And why to thank progesterone? Well, because progesterone is doing everything. Progesterone is sending message that, okay, blood vessels, you dilate. Yes, those decidua, right? By the time, say, these say, blood vessels, they are developing, right? You create all these, say, food packets and keep feeding them. So who was doing this, right? This sugar-rich fatty fluid, that is because of cells of decidua, all was because of progesterone, right? Because progesterone was knowing that uterine vessels, they won't be dilating, and those lacuna and those pools won't be created immediately. It will take some time. So that is the real food So this is like a real food, right? And that was when the decidual cells, they turned into their role of waiters, right, with sugar rich fatty fluid, and that was like a welcome drink, right? That was like a welcome drink. And they were so sincere that initially there were few cells, but as they knew that now the demand is increasing, so then more and more cells were deployed, so much so that those cells, they died. They created all those lacuna, right? Those lacuna filled up and joined together to create a large space. That is the space in which uterine vessels, they broke, and then finally this is the situation, right? Yeah, that's right. Coiling starts, all this is because of the progesterone, right? They will break because as they start dilating, after one level, right, their cell structures, they break. So that's where the pool is created, right? Okay, now starts the most interesting part. Most interesting. Till this point, right, this was the cytotrophoblast, and this was like the embryoblast, right? Let me change the color. This was like the embryoblast. Now starts the differentiation. See, it till this point though, it was okay. The real journey starts now. The thing what is called as the differentiation. It differentiates, right? It differentiates. Who differentiates? We are now dealing with embryoblast, right? They will differentiate. It means they will change their structure and they will become two layer, right? It is what is called as the bilaminar embryonic disc, right? Embryonic disc. Technically speaking, it is just the rearrangement, right? So now they rearrange themselves something like this. There is one layer, right? And then there is other layer, other layer. So they just differentiate. Few cells, they become what's called as the, this is called as the hypoblast, right? Again, see, blast, because their function is just to create, right? And this one is called as again epiblast, right? Because again its function is to create. So this hypoblast is on the ventral side, and this would be on the dorsal side, right? So just two layers are formed. Uterus get nutrition uh, to make zygotes. Zygote is now no more zygote. Now to he is our, say, big one. Zygote was that initial one, single cell. Now though it has got so many nutrition, so many blood vessels, right? Those, that pool, and through that pool, if you remember, I talked about syncytial trophoblasts. Those syncytial trophoblasts, they are creating those primary villi. So those primary villi, that is where they will be later on, there will be secondary villi, and in secondary villi there will be development of those small delicate vessels, and these blood vessels, they will be communicating when the heart starts beating, right? We'll come to that, right? So right now, just keep it that much, that the stage is ready, right? The stage is ready. Nutrition is for developing embryo, not for the, yeah, that's right. Because uterus so is as such getting nutrition, right, from its own arteries. These are the specific arteries which are getting broken, right? And for the vessels, for the walls. Okay, now coming back to this bilaminar, right? Bilaminar, that there are two layers. So one is hypo and second is epi. This hypo, anything which is hypo as such, his future is not good, right? Because he is hypo, right? His future is not good. This epiblast, his future is very good, right? So let's see how their future, right? What they now do is, now on every page I'll create one figure. Lacuna is actually not releasing anything, because lacuna, these are the spaces, and in those spaces, right, that blood will start pooling up. And then that blood, right, it would be collected and it will form a big pool, right? Those lacunas, they connect with each other and they are filled with fluid. That's what I said. Lacuna means space, right? And in that space, because if you want to put blood, so then there has to be some space. So those lacunas, those spaces they are created, they combine together, and then the blood will be going inside, right? That blood would be collected and it will be giving the embryological nutrition. That's right. Yes, through lacuna, embryoblast gets nutrition. Perfect. Perfect. Okay. Now, on every page, what I was telling, that I'll be creating one figure, right? Keep on visualizing that what is happening, and the moment I draw, it is completely fine. You speak off, right? You keep speaking that what is the name of that particular structure. That way, all the way will be going to from 2 layer, will be going to 3 layer, from 3 layer will be developing that notochord, right? Everything. But you keep orienting yourself properly, right? So the best thing is keep speaking that what I am drawing. So when I say this, so this is cytotrophoblast, correct? So that is cyto. Now I'll not draw all those individual cells. We know that there are all those flat cells, single nucleated, everything, right? So this is cyto, and capital T would be now trophoblast. So wherever trophoblast will come, we'll just write capital T. Okay. And we do have 2 layers, right? Now we do have 2 layers. Now for both these layers, we'll use a different color, right? Let's use a different color. One would be yellow and one would be this blue. Okay, right? So here it is, first our blue color, right? This blue, because it is epiblast. So that's like this, right? And it differentiates and it creates a cavity, right? So this is epiblast. So first there was a cluster, then that cluster became 2 layer, and then this formed one balloon. This formed one balloon, right? A chamber. So there is one chamber, there is second chamber. One chamber which we have already drawn is epiblast, right? Epiblast. So that is the chamber which has been created by epiblast. Now, obviously, here is some space created, right? There is some space created. So we'll call this space as amniotic cavity, right? Amniotic cavity. Yeah, that's right. From inner cell mass, epiblast and hypoblast they are formed. So what, any query? Not from the trophoblastic cell. Yeah, that's right. Right? Because cytotrophoblasts, they are outer, right? Then from that over here, this one, right? This is what was embryoblast, correct? Or you can say inner cell mass. From that, bilaminar disc. Right? Hypoblast, we have not drawn it. It is there. So this is the epiblast and this is amniotic cavity. Meantime, here it is, right? And this one is hypoblast, which you are missing so much, right? So now you'll be happy. This is the hypoblast. But unfortunately, very soon hypoblast has to go, right? But doesn't matter, right? So this hypoblast, right? Here also there is something filled up. Now it is called as yolk sac, right? That is yolk sac. Now in Mumbai, there is one area called Church Gate. Neither there is church nor there is gate. So exactly the same way over here also, there is no yolk sac, but the name is there, right? So this is what is called as the yolk sac. In fact, it is filled with vitelline fluid, thin fluid, right? It is filled with vitelline fluid. That's what it is filled up with. And this fluid, its function is nourishment, right? Nourishment. It nourishes that embryo. So here it is that hypoblast, epiblast, and everything is fine. It is this epiblast which will be playing very important role in formation of 3-layer disc. So now from this point, we will make it a 3-layer disc, right? How come? So that's the first one. That is our cytotrophoblast, right? And then it was blue color, right? Blue color, yeah, blue color. So, so this is, now I'll be drawing it bit fast, right? So this is epiblast, and from epiblast there would be transformation. That would be the differentiation, and that differentiation is very interesting. It is what is called as the extra embryonic mesoderm, right? Now what suddenly this such name has come? The thing is, this will go out, right? It will say, I'll be remaining out. So that's why it is extra embryonic. So from this epiblast, few of the cells, they will start differentiating, and they are now extra embryonic, because embryo to is inside, right? This is going on to the outer wall, right? That is extra embryonic, correct? Meantime, this will remain, right? That is already there and you know it, right? So this would be like hypoblast, right? It would be hypoblast. This would, Y is yolk sac. This is amniotic sac, correct? And that's what you have to remember now. This is extra embryonic mesoderm, extra embryonic, because the central portion, this one is the real embryo where the entire game is getting played. So rest of the things, they are out, right? So that's why it is extra embryonic. Okay. Now this lining, which is inside cytotrophoblast, right? Am I right? That this lining, it is inside cytotrophoblast, because that white line is, now we have not written, but it is like cytotrophoblast, correct? And it is this lining which later on lead to chorionic cavity. So right now we'll not be dealing with the chorionic cavity, but it is this, later on it will lead to chorionic cavity, right? Because things will be dividing and will be creating a cavity. Okay, so for 2 weeks, up to week 2, this is all what really happened. Now we are entering into the week 3, right? Third week, right? Week 3. Let me save this file. Done. Okay, so now week 3. Where is the blastocyst? Why such question, right? Because you tell me, where is the blastocyst? What was blastocyst? Right? What was blastocyst? Blastocyst, gentlemen, was all the way when we were talking about, right? All the way. This, if you wish, you can, right? When it was the cytotrophoblast, when it was trophoblast, then there was embryoblast, and then this was like blastocele, and this unit was just inside unit was called as the blastocyst. You remember that? That was the blastocyst. After that, the blastocyst, he has developed so much, he became stylish, he became sensational. Syncytiotrophoblast came into picture, and then that inner mass cell, it divided, right? This was like, right, what you are watching over here. You can say that this is the blastocyst. But well, over here we have not drawn syncytiotrophoblast just for the sake of simplicity, right? That blastocyst, so that is now the time of the past. Now he has developed, he has, from the blastocyst, he has developed epiblast, hypoblast. Then there is yolk sac amniotic cavity. On top of it, it developed this extra embryonic mesoderm. right? That and extra embryonic mesoderm. Now we have reached to the week 3 and see what happens, right? Say it happens like that. Sometimes if you get lost, don't worry much, right? This is a sequence. At times you, and that's why this file would be given to you. You might have to watch it 3-4 times so that till it sets in the brain. The most interesting thing should be you start taking 3-4 color pens and just draw 10, 12, 15 pages, or if you want to draw it on iPad or on your laptop, anywhere, just keep on drawing. When you'll draw, when you'll keep on marking, things would become crystal clear, right? Okay, so it is completely fine that sometimes it happens on the first stroke, suddenly it becomes difficult. And now say we'll be developing the primitive streak. It will become still more complicated, because that's the point from here. This week 3 is the most important, because now there would be migration. You have to actually think in 3D, right? You have to actually think in 3D that how exactly it is happening. Okay, so beginning of week 3, right? Cytotrophoblast, and then I'm drawing just a simple line. Oh, sorry. Let me take another color. Yeah, this is just a simple line, and from the color you should be able to say that this is extra embryonic mesoderm, right? And then we are keeping the blue line. We are keeping it for that epiblast, right? And then there was a yellow color which we kept for hypoblast, right? So even if I have not labeled everything, still I hope you are with me, you understand, right? Now let's put the direction. Which is head end, which is tail end? Who knows? Who knows? Right? There is nothing like we can really figure it out. We just know ventral and dorsal. We just know anterior and posterior, because this hypoblast, as it was said, that hypoblast is on the ventral side, right? Hypoblast is on the ventral side, and on the dorsal side we have got the epiblast, right? So far it's fine, but which is the head end, which is the tail end? No, we don't really know that. Now starts a small portion of, see, the entire work is by epiblast. Epiblast is everything. One portion of the epiblast, it starts multiplying. One end it develops, right? It multiplies. Yes, immediately it is said that okay, the end where it is developed, it is called as the caudal end or the tail end, caudal end, and opposite end would be the cranial end, right? So it is the epiblast who decides that wherever it starts multiplying, this, so it is a caudal end, and this is what is called as the primitive groove. Primitive groove, right? Now what primitive groove is, right? Primitive groove. See, primitive groove, it is nothing but, as we said, that at one point of time, one end, it starts replicating, it starts dividing, right? And that is where the head and the tail end is defined, right? Head and tail end is defined, because as we said that it would be only from the caudal end or the tail end, right? And now this unit, right? This unit is a special unit now, right? Till this point, though, it was okay, right? Everything was same, but now we need to give it a name. We should give it a name, and that name is called as the gastrula. Gastrula. A new name, because see, he has decided that which is the head end and which is the tail end, right? So we need to give some name. So that is gastrula, and that's why this process is called as the gastrulation, right? It is gastrulation, gastrulation. So that's the process of formation of gastrula is gastrulation. Say head will be down, say, say this orientation. This can be any orientation, right? It can be in any of the, this is for our sake of simplicity. Otherwise, say if you see in 3D, so it keeps on moving in any of the direction, right? But wherever that cluster develops, that is the tail end, right? So right now, don't think like that because the baby's position is like this, right? That's why it is locked. No, it can be in any of the orientation. This is for understanding. Okay. Now, once this is day 14, right? So we are on day 14. 2 weeks have passed. The third week has started. So let's say it would be day 14, 15, right? When the gastrulation process and the tail end is created. Now, if you really now watch it with two perspectives. One, you're watching it from the top and you have to watch it from the side, right? You have to watch it from the top and watch it from the side and think it in 3D. Now this time it is not like a flat structure. Now it is like a ball, right? It is like a ball. When it is like a ball, right? So now everything is epiblast, right? Epiblast is main. So now think it that as if you are watching from the top, right? Top. Till this point, you were watching from the side. So on the side, this is like watching from the side, watching from the side, right? So that there are two layers. But let's say if you are watching from the top, right? Just you are watching from the top. So then you are watching only the epiblast, correct? So here it is that cytotrophoblast is everywhere, right? And then there is extra embryonic mesoderm is also everywhere, right? They are just complete ball. But on one side there is epiblast, on other side there is hypoblast. So I am watching it from here. So I am just watching the epiblast. So this epiblast, its shape is now like this. Previously it was all same, but now its shape is like this. This is the shape. This is what I want to tell. That previously its shape was like this. Sorry. It was like this. It was like this, round, right? But now its shape has become like this, exactly, like this. And yes, there are all cells, right? There are all cells. And this primitive groove, it got formed here, right? So in top view, this is primitive groove, right? Primitive groove, right? Rest you know. And this would be like then the cranial end, and groove is indicating that it is the caudal end, right? And rest all you know, cytotrophoblast and the extra embryonic mesoderm, extra embryonic mesoderm. Yes, yes, yes. Extra embryonic, say, layer is another form of epiblast only, but differentiated. When we say differentiated, it means it's a mesoderm. Now it has changed, right? We cannot call it more epiblast. Another thing, how we able to see epiblast in the dorsal side? So from top, how come we'll be able to see the dorsal? No, 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 no. See, watch it in, think of, this is a football, right? This is a football. You divide the football into two parts, right? So two sides would open. Now, take it that way as if it's a, forget football. Take it like it's a watermelon, right? So watermelon has got the outer that sheath, which is thick, and inner core, something what we eat. Now, what if one portion of that watermelon is spoiled, right? So will it be, say, if this is the watermelon, right, and that one portion is spoiled, right? When I'm drawing this thing, this is in 2D, correct? This is in 2D. But when you take the full watermelon, right, and let's say the full watermelon, and this is the portion which is not very good, right? There is something like something bad, right? It is spoiled. So this portion, if you really want to see, so you can see this thing in, let's say this is the shape. So you can see it one from the top, right? So when you are watching from the top, obviously other structures, right? Other good watermelon will be there. But just for the drawing purpose, it is like if I take a section like this, so I'll say that this spoiled portion is round, right? So it is in entire football. But when I take a section like this, so you'll be watching only this portion, right? So one is watching from the top, second is watching from the side. That's what we are doing. Boiled egg is best. Yeah, boiled egg. I don't eat eggs, so that's why it never came in my mind. But well, you are right. Hypoblast? No, no, it has not gone anywhere. It is there, right? Hypoblast is there. That's what I said. We are watching it from this side, right? We are watching it from the this side, right? So when I say the top view means this is like a ball, right? This is like a ball. So we are watching from that point where we can see only the, we can watch only the epiblast. If we go on this side, right, on the opposite side, so yes, we'll be able to watch only the hypoblast. True, right? But hypoblast is least important because he's hypo, right? So he's going to die very soon, right? So it is this epiblast will be playing good role. Okay, so let's move further. So we are now watching the epiblast only, and in that epiblast, wanted to emphasize that this is the shape. This is the shape, right? So when I say top view means not actually, because there is nothing like top, bottom, nothing like that. It is this point, right? Or let me remove this top, right? And put the arrow. Put the arrow, right? And this arrow is with reference to this. So I'm putting a star here and I'm putting a star here, right? Now it makes sense. Yeah. So no confusion, right? Yeah, third. So this is like on 14th, 15th day, right? 14th, 15th day. So over here, that's the shape and the primitive groove is formed. Once the primitive groove is formed, so the tail end is now finalized. Now if I take that section, this section, right? This one, right? It is like that we are just taking, it's a say orange, and we take exactly the middle section, so we see all the pieces. So same way over here. Now it is this what we are about to watch over here, right? So that's what I'll draw it again. So here, once again, cytotrophoblast, and this is the extra embryonic mesoderm, right? And for understanding, I'm just tilting it 90 degree because as such this thing keeps moving. So here it is what it is, right? The color, it is epiblast, right? And this yellow color, that is the hypoblast, which you are missing so much. Right? So hypoblast. And what is inside? Yes, you are right. That's yolk sac, right? Yolk sac in which there is no yolk. And here it is. This is epiblast. Now this epiblast, it created that streak, right? But then it keeps on moving and it becomes bigger on the head end. So for the to start with, it was at the tail end, right? And then it becomes bigger. So it is like a continuous process. And surrounding it, all those, no, not this color, right? It was all that syncytial trophoblast, right? That is over there. We want to emphasize on these points, that this is the cranial end and this is the caudal end, So that was the beginning of that primitive groove, right? That was the point. That's where it started. Okay, yellow color is attracting, but that color is about to go, right? Okay. Now focus only on epiblast, right? Only on epiblast. So that epiblast in it goes like this, right? That epiblast, so from the posterior side, right? It is primitive streak, then it grows further, and then on the anterior side, it develops a node, right? Prominent. Then there is a depression in that. Then there is a depression in that, right? So I'll draw it like this, like this. So let's name all of them, right? So over here, it was like that, right? This was what? This is cranial end. So it was primitive groove. Exactly, right? It was primitive groove. So here is the primitive groove, right? But primitive groove, we can't watch it from the side. We have to watch it from the top. But this would be the location, right? From side, we can watch this, right? That prominence. So this is what is called as the primitive node. Makes sense, right? It is prominent. And then there is a dip, right? In between like this, right? That pit. So this is what is called as the primitive pit. So primitive node, primitive pit, and primitive groove. All of those combined collectively, we call this primitive streak. Primitive streak. Remember, this is all what is happening in epiblast. Now see, the epiblast is becoming more and more prominent, right? It is working so good. Second and third subject. Right now, don't ask about other subjects. I'll answer it. Right? I said I'll be teaching so many subjects, but right now, don't ask about anything else. Right? Just enjoy this. Right? We'll talk about all other matters later. Okay, so that is primitive streak. Now see what this primitive streak does. Right? It looks so simple, but what it does. This is the point where you start thinking in 3D, right? Think it like this, right? First, I'll not draw. I'll just explain, and then I'll draw. This is the ball, right? And over here, this is the epiblast. This is the hypoblast, right? They both are like this. Now, let's make it ulta. Why? Just for the understanding, because over here in the mother's womb, this is like a space, right? In space there is nothing seedha, nothing ulta, right? Every direction is correct. So for understanding, we can definitely understand it like this also, but then as I want to explain you that 3-layer, right? So that's why I'm making it this ulta. So let me draw. So once the image is also in front of you, it becomes much easier. Cytotrophoblast, extra embryonic mesoderm. So crazy, right? I'm making it so. And epiblast, because it is important, so that's why I'm drawing it above, right? And this is, right? Watch this hypoblast for the last time. He's about to go. Okay, so this is the situation. Now, first thing happens that this epiblast, right? Some cells they start migrating, and then they say that this hypoblast bahut hua, right? This hypoblast is now not needed. Let's take over. So then first step occurs that this epiblast cells, right? they start migrating, and then they take over the entire hypoblast. Gone. So this is when your epiblast, right? I'll write the keywords. Epiblast differentiates, right? Differentiates. It means it changes its form. Migrate down, right? It will migrate to hypoblast, right? Hypoblast, and takes over, right? It replaces, literally it replaces. And this new formation is now called as one of the layer, very important layer, and that is endoderm, right? This is endoderm. Obviously, this is embryonic endoderm because still everything is in embryo, right? It is developing. So we say embryonic endoderm. Got the idea? So that epiblast, right? He was keeping an eye on this hypoblast that let me get a chance and I'll replace you, right? The moment it came, right? Those few epiblast cells, right? They took over that entire hypoblast. Hypoblast is gone forever. Goodbye. Thanks for services. And it's over. And now this area is called as the endoderm, right? This entire thing is now the endoderm because epiblast has differentiated. So this is the first point. What happens? Epiblast would say, no, things are not yet over. Few of the other enthusiastic cells, they'll say that, okay, we'll not dive that deep, but what about this space which is in between? So they'll migrate and they'll migrate and they will connect with extra embryonic mesoderm on both the sides. So now the area which is in between over here, right, which I am drawing, it is not extra embryonic, it is not outside, right? It is not. Role of hypoblast was just to give that chota mota nutrition, right? As soon as its work is over, right, they have kicked him out. So it was giving that minor nutrition, right? And at times it happens like all these spaces, they are giving a form. And then when the differentiation starts, right, rest of the cells, they start giving way. Same thing, even in this also will happen. Okay. So that mesoderm, see now the mesoderm is the smartest person, right? What mesoderm? Still there is one more very smart movie is about to come, right? You like it. So this mesoderm, this mesoderm is embryonic mesoderm, right? So see, the names are not that difficult. We'll call this as the embryonic mesoderm, and this is extra embryonic mesoderm, correct? One which was outside and one which was this inside. So this is embryonic mesoderm and the extra embryonic mesoderm, they fuse, right? They fuse. And finally, so that is the second point. This is the second point. And finally, the third one, right? That is who remains as it is, right? Who is not doing anything, and this whatever is left out, right? He will be the ectoderm. He will be the ectoderm. So these are like ectoderm. This one. So this one is the third one. So that way, if you see now we have got the 3-layer disc. It is what is called as the trilaminar disc, right? So we have got this ectoderm, embryonic, right? So all are embryonic ectoderm. Only the mesoderm, it was the extra embryonic mesoderm. In fact, I tell you, in case of extra embryonic mesoderm, right, that also divides. That also divides into 2 layers, right? That is parietal and visceral mesoderm, right? Okay. So 3 layers, ectoderm, mesoderm, endoderm, they have been created. The result is trilaminar disc, right? Now this is ecto, meso, and endo. Many times you will use you'll find the word germ layer. No difference. It is absolutely the same, right? So when we say trilaminar, in which there is ecto, meso, and endoderm, right? Do remember that they are also called as the germ layer. These cells, they are multi-talented cells. So that's why we call it multi-potential, right? As such, the word is multipotent, but I always add this multi-potential. They have got so much of potential, right? They are so much talented. And why we say that they are talented? Multipotent means they can differentiate. To differentiate means to change themselves, right? You must have seen that, right? These cells, they change themselves so much. So they can differentiate into any organ, any tissue, right? They can differentiate any organ or any tissue. So that's why entire body is created from this ectoderm, mesoderm, endoderm. This is the reason we were telling right from the beginning that this epiblast is the hero, right? He'll be taking care of everything, right? Okay, now we have reached to the day 15, right? Day 15, 15, 16, right? Some super interesting thing happens. 1, 2, 3, right? Ectoderm, mesoderm, and endoderm, right? Ectoderm. Now these three layers, right? Mesoderm is not touched. The ectoderm and the endoderm, right? They meet. Ectoderm and endoderm, they meet. This is cranial end. Cranial end. Mesoderm is completely spared, right? So just 1 and 2. So this is what is called as the cranial bilaminar, correct? 2 layers, bilaminar. And this is the point which will later break, and this will lead to formation of mouth. That is on the 4th week, right? You will see that the mouth would be formed. And that's why this particular layer is very sensibly, it is called as the oropharyngeal layer. Nice. On the posterior side, right? On the other side. So this would be like cloaca, right? That is the caudal. So this is like a tail end. Caudal bilaminar layer. And this caudal bilaminar layer, that will lead to formation of anus and genitourinary tract, right? Genitourinary tract. And that is bit late, right? Roughly about week 7. So that's how it is formed. Yeah, this is also called as this junction, cloacal membrane, right? It is also called as the cloacal membrane. So that is the membrane. All these things, they are very vital because many abnormalities, right? When you'll study, immediately you'll understand that, okay, what if this layer, it is not broken, it sustains, or if there is any change, right? All those abnormalities, they will develop from it. But this is one important point, right? Oh, we have overshoot, but still just 10 more minutes, right? We are almost on the, say, day 15. So this happens for 2 days, and there comes the day 17. Now day 17 is a super intelligent day, right? Very intelligent. Why so? That know this, right? Know this, correct? And say now we can draw this, right? This also, you know. And this, right? And in between this. So we know that this is extra embryonic mesoderm, this is embryonic mesoderm, and this one is ectoderm, and we have this as the endoderm. Okay. So in between there is formation of notochord. Notochord, right? This notochord is like a transient thing, right? In human life, right, there is nothing much which is made up from notochord. In fact, it's like a transient structure, it goes away. Only some part of it remains in the formation of some portion of that nucleus pulposus, right? There is one intervertebral disc, and in between, right, there is a nucleus pulposus which is soft. That part of the nucleus pulposus is like remnant of this notochord, right? So I'll just say part of nucleus pulposus, right? It is just a remnant, right? Rest of the things will go away, but at this point of time it plays a very super smart role. One, this notochord is a tough structure, right, which is made from this mesoderm. So it is tough, it is solid, right? It is solid structure, so it obviously gives the direction, right? When this entire, right now, so it is, everything is like a ball, then it will start elongating, then it will start folding. So it is at that point, it is notochord who will help in folding. So it helps in folding, right? So that is when we'll be studying, say, further third month, fourth month. That's when all those things occur. But the very interesting thing which is over here, it is notochord secretes something, right? It secretes SHH. SHH, this is a protein, right? It is SHH protein. What SSH? Nothing very sensational. It is like sonic, right? Hedgehog, right? HEDGEHOG, sonic hedgehog protein. And this protein, it releases to everyone, right? So let's say it releases to everyone gets it, right? Don't worry, I'll remove this, but that's how it is released, right? And it is released that those who are near, give them more, right? Those who are far, give them less, right? Those who are far, give them the less. What's the importance of such thing? The importance of it is that its concentration is high when structure is near. So to near structures, concentration is high, and vice versa. So that way every tissue understands, and it is what is called as the 3D orientation, right? It will give 3D orientation to everyone, to all. And now they know that they are, how far they are from notochord. So that's how the entire 3D orientation comes into picture. And this leads to a very important developments, right? See how smart the things are. How smart, because this SHH, it is released to all three layers, right? Endoderm should be embryonic, because finally is differentiated epiblast. Endoderm is embryonic, right? We never say it is extra embryonic. Endoderm, ectoderm, and mesoderm, all three are embryonic. This is embryonic, this is embryonic, this is embryonic. Something which is out is extra embryonic. It is this, right? Got it? Because we never said that any of these three layers, they are extra embryonic. Means how can they be? Because this middle portion is the embryo. That is where things are happening. So how the mesoderm, how the endoderm can be extra embryonic? It is not. It is all embryonic, right? Okay. Now we move on to, say, this process. This process, it takes good 2-3 days, right? Now that way we now reach to the day 20. right? We are about to reach to the end. So over here, we just zoom this thing. This is notochord, right? So then this notochord, it tells the surrounding mesoderm, right? That what you guys are doing, right? Come on, differentiate. So these sleepy mesoderms, right, which were like this, seedha simple, right? But this notochord tells, no, don't do this. Develop, right? So then they develop, right, like this, like this. Similarly over here, like this, like this, like this, right? That way they both develop, right? This, remember, we are dealing with mesoderm. So in this, if you ask, it is this portion. It goes like this and this. It goes like this and this, right? But I'm removing this from here because it is day 17. Actually it happens on day 20, right? So remember, this is mesoderm, right? Mesoderm. Though we have used white pen, but this is all mesoderm, right? It is mesoderm. It is mesoderm. And this mesoderm has got a name. Simple name, this one, because it is near to the axis, paraxial, right? This one is lateral, right? You know why. Lateral is lateral. And in between, there cannot be better name than this, intermediate. Right? So this is intermediate. So these are the various mesoderms. So next time, when in any of the embryonic development, the moment we say that this is what we are dealing with, it's a paraxial mesoderm, immediately it should strike that yes, it was a notochord, and who said that go ahead and become, say, this paraxial. So paraxial, intermediate, and the lateral one, right? Another important thing, this notochord is so influensive. Notochord on day 20 only, right? On that day only. So many smart things are happening on day 20, right? Still we are on day 20. Notochord tells, okay, I'm sitting over here, right? Who is above that notochord? Ectoderm, right? Just for the reference, here it is, right? Here it is the ectoderm. This is ectoderm, right? So this notochord tells to ectoderm also, you also do something. So he'll say that, okay, I'm like this. He say, no, this is not good, right? You just, this above plate, right? Whosoever is right, just above me, that plate is thickened, right? That plate is thickened. And he'll say, not only this way, right? There should be some style in that, right? So he'll say, okay, should I form a groove? He'll say, yes, form a groove. That groove is formed like this. It goes like this, like this. And this plate, this is thickened, right? This is thickened. This portion, right? It is obviously ectoderm. It is thickened, right? And that thickened is neural plate. It is what is called as the neural plate. Name itself is telling neural. Yes, it will be playing very important role. But this neural plate is like, say, these two are prominent, right? They keep on increasing. So they take this shape, right? They become like this and this, right? From here. Then they will become like this and this, right? And then this is what really happens. It becomes like this and this is separated, right? Because when these two poles they meet, they both meet. So this is separated. And where is the notochord? Notochord is just here, right? Now it makes good sense, right? So that is the notochord. This is the standard ectoderm. And this was right from the beginning we were telling neural plate because it was flat. Now no, it is no more flat, so we'll call it neural tube, right? We'll call it neural tube, right? So that neural tube which is formed. Finally, the situation was that those syncytiotrophoblasts, right? Syncytiotrophoblasts, right? They are now, because of this much time, they are more connected, right? They are more connected, right? And then that extra embryonic mesoderm, right? Which was that extra embryonic mesoderm, which was there. So it migrates. It migrates to that, right? This is extra embryonic mesoderm, right? It migrates to primary villi, right? Because primary villi were what? They were just the projection, right? So this extra embryonic mesoderm, it travels and it has got ability to form small blood vessels, and they are now secondary villi. They lead to formation of secondary villi, and in secondary villi there are vessels, blood vessels, right? Small blood vessels which are formed. Now these blood vessels which are secondary, right? They differentiate and they lead to a system, and that is called as the villus capillary system. That's what is called as the villus capillary system. Done. This villus capillary system is what is that baby is doing for placenta, right? So this is the contribution of the fetus, right? He is telling that okay, this much I'll create, right? Rest mother will create, and that's how the entire placenta would be done, right? And when the heart beats for the first time, right? When the heart beats, it is this villus capillary system which will be full with blood, and then it will start, say, sending all those nutrition and everything, right? So that's how we reach to this point. From this point onwards, the moment you say that okay, let's learn the heart, immediately from this trilaminar disc, what happens that it would divide and it will start formation of this, right? From where will come is either syncytioblast, syncytiotrophoblast. We discussed it right from the beginning. Syncytiotrophoblast, they are like, I'm repeating it, there was trophoblast. Trophoblast, some of the cells, they differentiate. They become syncytiotrophoblast. Syncytiotrophoblast speciality, they are multinucleated, right? They are multinucleated, right? And those single nucleated, we call them cytotrophoblast, and they were single nucleated, right? And then these syncytiotrophoblasts, they divide, and then they covered that entire area, right? Then there were development of primary villi, so that blood was getting pooled into that, and then with the help of diffusion, they were getting nutrition. Then that extra embryonic mesoderm said that till what time you will start getting this nutrition that way, let's create our own system. So that's where extra embryonic mesoderm started creating those small vessels. Those small vessels, right, they were now called as the secondary villi. So these secondary villi, in which there were small blood vessels, and collectively it was called as the villus capillary system. Villus capillary system is directly connected with our own mesoderm. So when the heart would be formed, these vessels, they will be connected and done, right? Villi are the other form of syncytial, not how villi form. Yeah, that's right. Villi means extensions, right? Villi means extensions, right? So this was the thing. Do revise it. For any query, don't hesitate in sending a mail, or you can write in comments. Anything is completely fine. No worries at all. But do revise it, because, and let me save the file, done. So do revise it, Because once you will revise it, draw these figures. You have to be scientifically correct. You don't have to be artistically correct, right? Drawing every cell like this, right? So, so no need, right? But do draw, right? Do label so that it should be crystal clear in your mind, right? I think we have overshoot quite a long, but so nice of you that all of you are still here. 2 or 3 second or third year subjects, surely, why not? Definitely, definitely. Oh yes, and in fact they will come very fast, very soon, because our plan is next week physiology, then the biochemistry, then we'll be going for pharmacology, right? Yes, sure. Will capillary will help in blood transport to heart or placenta? Well, it will be giving blood transport to heart via placenta, because placenta is the connection between the fetus and the mother, right? So that's from this point onwards comes the fetal circulation. Chalo, let's do one thing. You, as you have asked, this is a good question, right? In next week, right, wherever it would be possible, from this point onwards, I'll pick up and will see the fetal circulation, right? Fetal circulation, and at that point, I'll also take the development of heart also, right? So at least that portion would be done. The moment you will learn the heart, development of heart, it will become very easy for you to understand all the anomalies, right? So sure, we'll go for it. Thank you so much, right? I think even you have not taken your dinner and even I have not taken my dinner. No problems. Thank you so much and wish you all the best for tomorrow's test, right? Don't worry, test would be very easy. And it is like you are just evaluating yourself, right? Thank you and good night and take care. See you. Bye-bye. Thank you. Just save the file. All right. So this file is saved. Within 2 minutes, it will be landing into that folder, right? And from where you can download it. Take care.